Thank you for coming here. Just I'm giving a brief introduction about you. And
Yeah,
very good morning. Very good morning to all the attendees. And today we have our distinguished speaker, Dr. Bhim Pratap Singh, who is working as Associate Professor in National Institute of Food Technology, Entrepreneurship and Management, and that is in Sonipat, Haryana. And he's basically is expert in the area of microbial secondary metabolites, plant microbe interactions, and he has completed his PhD from Bundelkhand University with NVIM ICR institution, and has completed PDA from University of Aberdeen, Scotland, on microbial natural products. He is experienced experienced more than 15 years, and he is teaching BTech, MTech, PhD, MSc, BSc students since last 10 to 15 years. He has been awarded and honored by many fellowships, and also awarded his Best Oral Presentation Award at National Symposium held on ICR, Indian Institute of Pulse Research, Kanpur. He has received Innovator of the Year award in 2017 by Scientific and Applied Research Center, Meerut. And he has got Best Achievement Award from Contribution to Asian PGPR Society. And also Young Scientist Award he has been given by Association of Biotechnology and Pharmacy. He has many international national research papers published in many reputed journals. More than, I think, 70, 80 research paper he has been published till yet in many many reputed journals with having high impact factor value. He has international experience and also collaboration and consultancy experience in many DBT related projects, and he is selected as DBT Overseas Associateship for 6 months in University of Aberdeen, Scotland. And he has got many patents, more than 7-8 patents he has been awarded, and he has published so many books, more than 6 books he has published till yet. So he is having very vast experience in the area of plant and microbial interaction area. So we will learn many things from Dr. Bhim Pratap Singh sir. So now I am inviting him to deliver his talk because we are waiting from yesterday to his lecture. Welcome, sir.
yeah. Thank you very much, Professor Kamlesh sir. And I am really thankful to the authorities of AKS University, who has given me a chance to talk to the attendees and the participants. And yesterday, actually, I was traveling back from Ranchi, so I could not make it. And on my request, Dr. Kamlesh was so kind that he has postponed the talk. And today, I will be sharing some of our experience which we had while working in the last several years. So if you see, the topic which I have selected is a somewhat different topic, which is about biofortification and agriculture, how this particular group of endophyte is useful in agriculture. So when we talk about biofortification, and before I start my talk, I'd like to say that I was working as assistant professor till last year in Mizoram University. And this year, beginning, I have joined this organization. This is a National Institute of Food Technology, Entrepreneurship Management, which is in Sonipat, under Ministry of Food Processing Industries. It's a very high-tech institute for the people who are interested in food processing industries or food processing. We have a big pilot plant facilities where we can do the processing of X number of foods, and we can make several products. So this is more applied aspect of the basic work what we have done in Mizoram University. So I'll try to present. I'm presenting the work which we have done in Mizoram University basically, because here still I am in the initial phase. So we are starting up some of the works. So now when we come to the fortification and agriculture, So the term biofortification, I think many of us knows the meaning of fortification. Fortification, when we talk about fortification, we talk about the processing of food, that is nutritional quality of the food supply. When we talk about fortification, this is the practice deliberately increasing the content of an essential micronutrient. So fortifications is basically for the increasing the concentration of micronutrients. We are not talking about macronutrients over here. So as you all know that fortification foods are nowadays are available in market. We have lots of fortification foods which are very, I say that fortified foods can be beared by the rich peoples. But when we talk about the common people, fortified foods will be difficult task for them to procure, to buy, because fortified foods, by making the processing of crops, by making the processing cost, it becomes a very costly food. So what is the alternative for the peoples? So a term has come up. It's not much work has done on biofortification when we connect it with agriculture. So when we talk about biofortification, it is the nutrient, increasing the micronutrient level in the crops. So we can talk about application of some bacteria, fungi, actinomycetes, actinobacteria, how they can be applied in the crops on the soil, and how they can be helpful in increasing the concentration or amount of these micronutrients in the crop itself, so that from the crop it can go to the fruit and it can get a good, biofortified end product which can be given to the consumers. So some of the examples are there, which the projects which are coming up in biofortification are iron biofortification of rice, beans, sweet potatoes. These projects are going on. I mean, iron, how to enhance the iron content in these crops. Zinc biofortification in wheat, rice, beans, sweet potatoes, and then provitamin A, carotenoid biofortification of sweet potato, some amino acid and protein biofortification of sorghum and cassava. So these type of projects are going on. So I just like to what I have tried to explain that what is the importance of biofortification and why we need to worry for biofortification. That if we want to supply something to a common people, presently there is this conventional breeding and agronomic biofortification and genetic modification are some strategies which can be adopted to produce biofortified crops, because the biofortification is the only solution to alleviate the hidden hunger or malnutrition at this moment. So if we need to alleviate the malnutrition, biofortification could be a green solution or could be a sustainable solution by which we can alleviate the hidden hunger or malnutrition. So I'll try to explain with an example of endophytic actinobacteria, which we are working on, what is the applications of this particular group for the crop improvement. Oh, this is few of the photos of my present organization. If anyone of the attendees is interested in working on any of the entrepreneurship development in the field of food processing industries, he or she can look into the website, and you are always free to contact us. We will try to help us as much as possible. So what are the challenges? As I said, food for growing population. By 2050, we are going to have around 9 to 10 billion of peoples on the earth. So definitely there is a need to do some strategies to increase the biomass production, because land is limited and we need to see how the biomass or the crops can be enhanced. So malnutrition or hidden hunger needs attention. So malnutrition and hidden hunger needs attention means when we talk about malnutrition, nowadays malnutrition is very limited due to the availability of food. So what is the definition of hidden hunger? Is the not availability of micronutrients in the food. So if you have a huge amount of food, but it is deficient of micronutrients, then also you are going to have a malnutrition. So there is a need to understand the usefulness of these micronutrients, how we can fulfill the micronutrients quantity of amount in the crops or in the food, because micronutrients are playing an important role. So now another aspect of this crop production is we need to tackle, when we entered into the food production, food processing side, we understood that we have in certain crops we have a sufficient amount of production, and while it is lacking the processing part, I mean post harvest of the produce is the lacking part to many farmers or several places. So there is a need to understand or to establish processing units so that there will be less crop loss. Fruit loss reduction is less costly than the equivalent food production. So if you see the statistics of economic statistics, then it is well understood that if we can preserve the food which is already produced, that will be less costly than the food production cost. So FAO estimate says that around 40% of the food produced in India is lost or wasted every year. So biofortification could be a sustainable and cost-effective solution to provide for this particular hidden hungers. Okay, so now we are working on endophytes. Now when we start and working on endophytes, we thought that why we should work on endophytes. So what we understood that it is a cost-effective manner, that we need not to have a fighting with the rhizospheric microorganisms. As you all know that rhizospheric microorganisms has a huge competition, and the soil microbe interaction, plant interaction is a huge complex process. So we are a bit relaxed with that process because we are not dealing with the soil microbe interaction. Rather, we are dealing with the inside organisms which are there in the tissues. So they are environmental friendly. We can use them in any form, anyway. Continuous and reproducible yields to scale up, as we know that microbes can be scaled up at any point of time. So these 3-4 arrows which are pointed out there, we are working on these lines. We are already started the work on plant microbe interactions for adaptation, stress tolerance, how the endophytes help in plant in stress tolerance, volatile organic compounds for agriculture, how we can use these endophytes for the production of VOCs, which can be used in the agriculture, and microbial metabolites for medicine, agriculture, industry. These endophytes have a great role in agriculture as well as in pharmaceutical industries. So we have came out with some of the secondary metabolites which we have purified, and then we are trying to do some further studies on that extracts. So these are endophytic group is bit less competitive as compared to rhizospheric population. So that was the reason why we have taken on the endophytes. Now a brief introduction about the endophytic microorganisms. Endophytes microorganism resides in the internal tissues and live as either symbiotic or mutualistic association with their host plant without causing any symptom of disease. So we can say disease-free plant. We cannot say healthy or something, but disease-free plant we have to select to get the good endophyte. They are ubiquitous in nature. Present in all land plants which are there on land. There are several roles. So majorly they are the good producers of secondary metabolites which help plant in fighting or develop resistance against diseases, and they are offers several benefits to the host plant, particularly growth promotion and protection from pathogens, and that under diverse environmental conditions they help to communicate, to interact with the plant in more efficiently. Now if we see the plants, So this slide shows that if we start from bryophytes, this angiosperms, pteridophytes, gymnosperms, the researchers have already documented the presence of endophytes. So the colonization of these groups in the endosphere is vast, and each group of organisms, plants, they carries the endophytic population. Endophytes application in crop management. So there are certain, there is a concept which recently in last year White et al. Has demonstrated very excellently in the what they have said that when a plant is interact with, when the microbe is interact with the plant, at that time there is a cycle which is known as rhizophagy symbiosis, which they have called it as a rhizophagy cycle. So that rhizophagy cycle involved in the plant, and when the microbe interact with the plant, and during this things, the what is the major functional of the endophytes are, they increase the nutrients ability of the plant through from the soil through the microorganisms. They have an antagonistic effect against pathogen and pests. So they secrete certain secondary metabolites which are not allowing the pathogens to interact or come near to the roots. So pathogenic organisms will not allow to interact with the root hairs, and so it will not allow them to enter. So enhancing stress tolerance potential of plants and modulate the plant development. Overall, this population which is entering inside the plant, it is entering through the rhizophagy cycle, and rhizophagy cycle will help plants in several ways. So when we come into the work which we are doing, this is the overview of the some of the activities which we are doing in our lab from last several years. What we are targeted, we have taken some of the medicinal plants, because as you all know that Northeast India is having as unexplored habitats. So what we have selected the traditional medicinal plants which are being explored, which are not being explored heavily. So we have taken those plants and start exploring the endophytic population, basically mainly actinobacteria and fungi. So actinobacteria and fungi are the two groups which we are targeting at this moment, because actinobacteria has a vast applications in pharmaceutical industries, because they are the vital producers of antibiotics or secondary metabolites. The same case is with the fungi. So our object, we started with the applications of this actinobacteria in the pharmaceutical sectors, but later on through literature we came to know that these endophytes are also playing a good role in enhancing the crop disease resistance ability and for production of secondary metabolites and fighting against the diseases. So we have explored it, their role in the agriculture side. So how we usually do, we are taking a plant and then we isolate the endophytic actinobacteria. There are specific medias, so we can make it pure by repeated streaking. And when this endophyte is poor in the pure form, you can do the microscopic analysis, SEM photography, and all those things which is required to be sure that they are pure in number, and to make it confirm that they are the endophytic nature. There are certain protocols which we have followed. If anyone is interested, please look into the research papers where we have explained the protocol in detail, or you can just text me so that we can try to explain in detail. So then after we get a pure bacteria, then we have we can make it fermentation, and we can use in X number of applications, what as per the reported literature. We have also seen the secondary metabolite production like paclitaxel, choleotropic acid, and vinamide. I mean, some of the quercetin which we have in our collection, we have done the how much amount of these metabolites this particular bacteria is having, and then we can do for the PGPR activities which is related to the agriculture side. Then we have seen the PGPR attributes like production of IA, production of siderophore, extracellular enzymes, and whatever is you would like to try, we can just have a look whether this organism has these abilities or not. And if it is having, then you can go for identification of this particular bacteria, you can go for secondary metabolite production, you can go for biofertilizer development. So endophytes produce white secondary metabolites. There are some of the literatures which you can just for your reference. So we have seen that traditional medicinal plants are considered because they are good alternative source of plant-driven natural products. Based on this hypothesis, we have started working on traditional plants. There are several compounds. Until now, more than 80% of Streptomyces are good producers of several bioactive compounds. I'll not read the BPTs, this is just for your reference. So there are several commercial products available from actinobacteria. Now the question comes that actinobacteria is a slow grower. So there are several controversies on actinobacterial biostimulant developments based on actinobacteria. So what is the good point of actinobacteria is they are spore producers. They can sustain in the soil for a longer duration as compared to other bacterial population. So there are certain products which has been commercialized like Actinovate, Actinoiron by Streptomyces lydicus, and Argent for different strains of Streptomyces, Mycostop for stop the fungal pathogens, Blasticin S for Streptomyces from this particular Streptomyces, and there are list of products which are available. Now, what is the strategy? What strategy can be more good? I mean, more acceptable, as we know that actinobacteria are slow growers. So, what we are planning to do some mixing of actinobacteria and bacteria after seeing the synergistic effect. If they both are good with each other, then we can mix the actinobacteria, which is a spore producer and has the ability of PGPR, and the bacteria, which is not a spore producer but has a fast multiplication process. In that way, we are trying to make some synergistic effect of fast growing bacteria and slow growing bacteria. That hopefully that will work more better as compared to the actinobacteria alone products. So now this is all overview, and now let's just go for the methodology in detail. We have selected a targeted plant, purified the endophyte in pure form, did the identification, and then after it is pure identification, we know, then we have seen the literature, what properties is being reported this particular bacteria, then accordingly we can decide what side we need to explore it. So we can do the RT-PCR, qRT-PCR to understand that biosynthetic genes are there. If they are there, up to what day they are expressing in what amount, so that up to that time we can use for fermentation. And then we can go for identification, and then in vitro PGPR test. After in vitro PGPR test, if some strain is getting good results, you can always try for in vivo experiment by using the pot experiment. After pot experiment, you just want to understand what are the metabolites present in this bacteria. So we need to do certain chromatographic techniques. Either you go for GCMS or you can go for HPLC based any technique with the quantification, which can give you quantification of the known compounds. And if it is there, then you can do toxicity screening, and whole genome sequencing also can be done. Right, so one of the example which I am taking it, this is the work done and published already from our group some time back. So Mirabilis jalapa was the plant which we have taken. So now these are the all isolation part, how much actinobacteria we got from how many tissues. So for this there is a standard protocol. You need to follow the standard protocols while cutting the tissues, while plating the tissues on the media, how to do the sterilization, and all those are the standard methods. By following that, you can get the population of your interested bacteria from this particular plant, and then you can do some analysis, how much was in leaf tissues, how much was in the root tissues, and how much was in the stem tissues. And to prove that this is the endophytic in nature, you need to do all these things, that you need to understand the the population is coming out of the tissue, which is sterilized completely, and it should not have been come onto the plain plate where you have done the fingerprinting of the tissue. That means outside there should not be anything epiphytic in nature. So we need to do complete sterilization, complete tissue fingerprinting on a same plate, and then you need to do the same photography of the tissues itself, where you have to make a preserved one tissue on the plate and take it to the same studies. What you are seeing in the figure C, the root spikes, stem spikes or root spikes or root hairs, but somewhere in between there is a spiral type thing coming up. This is our actinobacteria. This is what, and then you can go in detailed overview of the purified cultures to understand that there is no contamination. And of course, you can use nowadays available tools, bioinformatic tools, to construct the grouping or to know the how many type of bacteria are there in this particular group, which are the rare one, which are the common one. In that way, you can construct the phylogenetic tree by using the mega softwares, or which is very much popular nowadays, and that can be done easily by just entering the sequencing data. Once the sequencing data comes, you can deposit it to the gene bank, and then once the gene bank accession is reached, and then you can construct the phylogenetic tree based upon the sequences. Antagonistic potential, we have a range of pathogens with us, agriculturally important pathogens. Now it depends here that which crop you want to target. If you want to target a tomato, you want to design some bioformulation and or biostimulant or your biofortification, something on tomato plants, you need to select the tomato pathogens. And then select the major tomato pathogens and see the effect on of this particular bacteria on that particular against that particular pathogen. So now it's your choice on which side you want to check this particular bacteria for development of any biostimulant. So you can make a broad spectrum of pathogens. Whatever is giving good results, you can go ahead with that in detail. I'll not read the values and all, so later on you can just read it. So what I will like to explain is that what are the different criteria or the attributes which you can check on. So IEA, ammonia are the two criteria which you can, when we talk about plant growth promoting attributes, I think most of you know that there are two mechanisms of plant growth promoting attributes. One is direct, one is indirect. So try to see the effect of this particular bacteria in both the mechanisms. So whichever is the maximum, maximum attributes showing good results by the bacteria, you can select that particular bacteria for further studies. So we have this Streptomyces thermocarboxylus, which is a good bacteria, and we have done the whole genome of this, and we have submitted this bacteria in NCIM. So it is there in Pune now. If anyone want to explore it for further studies, it can be explored by taking from them. So it's in the public database. So for doing this type of studies, this is also important that you have to submit your culture to some national culture collection so that it can be in the public domain after once you are done the work, so that it can be explored by other peoples. So we have done indole acetic acid. Now there is a biosynthetic genes. Biosynthetic genes means nowadays there is a primers for most of the attributes like phosphate-solubilization, there is a gene. IAA production, there is a gene. Indole, any whatever attributes, most of them, they are being designed. ACC deaminase, there is a gene. Primers are universal for most of the bacterial population. So what we need to prove is that we need to understand what bacteria we are taking forward. It is having that gene present or not. And if it is having that gene, that we need to amplify it, sequence it, and deposit that particular gene again into the NCBI. So sometimes please be careful that this type of biosynthetic genes, false amplification also comes. So false amplifications can be checked out after sequencing only. You cannot be very sure after getting a band of that particular size in the gel, but a back sequencing of this particular band and deposition of that particular gene is important in the database. ACC deaminase, yes, I talk about ACC deaminase already. So if there are several ways of ACC deaminase. It is a plate-based method is also available, and then ACDS gene is there, which confirms the availability of ACD deaminase activity. ACC deaminase is a gene or is an enzyme which breaks ACC into alpha ketoglutarate and the ethylene. So it suppresses the stress level of the plant or stress level of the plant when this particular organism enters into the system. Other plant growth promoting traits are phosphate-solubilization, extracellular enzyme productions like cellulase, amylase. Phosphate-solubilization, I like to explain an important part. Don't use always PKB medium. You have to understand the soil characteristics of your site first. If it is acidic, accordingly you can design the attributes. If it is basal in nature, please see the soil characteristics, whether your soil is required phosphate solubilizers or not. Right? Phosphate solubilizers are not recommended for each and every soil. So most of the PGPR attributes needs to be done after you have a detailed study of your soil. Use that specific organism for which there is a deficiency in the soil. Don't use blindly that phosphate solubilizers for X and Y soils. So soil analysis is the most important part. After you have a soil and statistics done, and then accordingly you select the bacteria to make it fulfill the deficiencies what are there in the soil. When you identified some potential strains, now you are going ahead for further experiments. You can do seed germination assay. It's a very easy assay for any variety of the soil which you seed which you are using. Just dip it into the suspension of this particular bacteria, and you can see which bacteria is giving how much percentage of seed germination as compared to controls. So these are the all standard ways mentioned all in our publications. Recently, this publication has come in PLOS ONE in March or January or something, I think November 19. So you can see this data if you want to go in detail. So this is the same way how we have done the in vivo plant growth promoting assays. What we need to do, we have different treatments of T0, T1, T2, T3. So what is T0 is the tomato plants grown in net house as control. T1 tomato plants grown in greenhouse with our interested strain of bacteria. T2 tomato plants grown in greenhouse with our strain and with the targeted pathogen, which is the major causative pathogen. And T3 is the tomato plants grown in greenhouse fungal pathogen with the pathogen alone. So here what we can understand that what is the effect of our bacteria in the presence of pathogen and without presence of pathogen. Without presence of pathogen, we can call it as direct PGPR. In the presence of pathogen, we can call it as a biocontrol effect. That this much ability is there in our bacteria to inhibit this much of sustain or resist against the pathogen. So while doing some normal statistics method, or by you can plot the graphs, and you can see the significant difference is there or not by using any statistical tool. You can see LSD, or you can even try for some variant analysis. Now, once this experiment is done, some certain physiological experiments are also required to further prove our concept that yes, when we inoculated this bacteria in the crop, in the seedlings, the root height and all those things have changed. This is a visually we can see, but internally physiologically what is happening inside the treatments. So we can you can estimate easily the chlorophyll A and B content, which can be done by the spectroscopic methods. So for the different treatments, you can do total chlorophyll A and B separate, so you can understand which treatment is having more chlorophyll content. So chlorophyll A and B was highest in our T1 as compared to treatment after 60 days when we grown the crop for 60 days. Further, we usually see the FB by FM ratio. This is a photosystem. This study is done in a specific instrument which we call it as doll palm. This instrument tells us that what is the ability, or how much is the electron transport rate and chlorophyll content. Electron transport rate takes place inside the treated and the control seedlings. So when we do this analysis, it can understood that how much electrons are get transferred to give more energy to the, or to do more photosynthesis. So this will give us, if it is high, it says it's good that FB by FM ratio is good as compared to control. So that we understood that yes, T1 showed significantly higher FB by FM ratio. Once we have done in vitro, in vivo, and then some physiological analysis, then we were sure, we were okay that the microorganism had something by which these changes are happening in the treated treatment in the T1. So now we understood that what is there inside the bacteria? What is there inside? Is there any specific metabolites, or is there anything which is we can correlate with the available literature? So for getting GCMS profile, we have done GCMS to understand the volatile organic compounds. We got some of the list of volatile organic compounds as compared to library. So out of them, we have seen that these 4-5 were already reported for their antimicrobial, antioxidant, and plant growth promotion abilities. So what we can tentatively say that, okay, these are the volatile compounds present in our bacteria, which could be responsible, which could play a synergistic role in the plant, that due to this, these activities are coming. So these are the few important compounds which we, in this way we can understand that with the reference literature, that these may be the responsible volatile organic compounds for this particular activities. Similarly, now this biofortification comes in. So biofortification means that when you inoculate the plant with your bacteria, what has happened in the plant? Is there some changes inside the plant or no? So we have taken the plant samples, did the GCMS of the plants also along with the bacteria. So we have taken the T1, T2, T1, T2, T0, the plants, and then send it for the GCMS analysis. And then we have seen certain some compounds which have increased, which are not there in the control, or which are different from the controls. So we can see that after giving the PGPR, this particular compound is more in production. So it is an antioxidant in nature, which was not there earlier. So there are certain compounds which are going more when we introduce our PGPR or when we introduce our strain. So we can say tentatively that this could be the responsible changes happening after introducing of our own bacteria. Detection, quantification, then non-volatiles we have done by the volatiles we have done with the GCMS. Now for volatile compounds or bioactive compounds, we have done the UPLC MRM technique. Multiple reaction monitoring is a specific technique of in HPLC, in which we need to have a standard, and if at all your standard molecule is present in the extract, then only the peak will arise. Otherwise, that peak will won't come. So this is a beautification of this particular technique, which we called as UPLC MRM method. So this is the result of this. We have got catechane, camphorol, paclitaxel up to this amount in mg per gram. So these are the few compounds which we quantified with the available standards with us by using UPLC MRM method. So as I said, the beautification is that only the peak will not come until and unless that particular metabolite is present inside the extract. So we have tried several standards. Out of them, we got some results which are being demonstrated over here. Then you can do the whole genome. As I said, we have done the thermocarboxylus bacteria. You can compare the proteome comparison with the available thermocarboxylus in nature or streptomyces in nature or in database. Sorry, not in nature, in the database. So with the comparison of this particular genomes, you can have more clear depth in information that what is there in your bacteria which is doing the role. So spermidine synthase gene was dominant in our bacteria, which is responsible for stress tolerance in the particular in plants. So spermidine synthase, which was there in a good amount, which is there in our bacteria, that can be correlated for this particular heavy metal resistance genes. We have got several heavy metal resistance genes in the Indian strain and some other strains which I have taken from the database. So our strain was having a quite good heavy metal resistance genes present in this particular bacteria as compared to the available data in the Patrick database. So in conclusion, what we can say that we have an endophytic actinobacteria which can be used as a bioinoculant for plant growth promotion, biofortification, and biocontrol agent against fungal pathogens. So Streptomyces thermocarboxylus has their application biocontrol agent in preventing and treating plant diseases in tomato, for which we have published patent, and the award is still pending. We have done the whole genome sequencing, and which is also suggesting the presence of genes responsible for these particular activities. Okay, so these are some other works which we are going on in our laboratories. I think if anyone is interested, they can see this type of work. Also, we are doing use of biomass to value added products. Mushroom biodiversity work is also going on, where we have done commercial production of one of the local mushroom of Mizoram, which we call as Rhizophyllum, and some of the projects which are going on in our laboratory there. Now I am not dealing with them, but some of the co-PIs, they are handling the projects. So there are some recent publications of our group, and I am sincerely thankful to the funding agencies, especially Department of Biotechnology. They have really funded our lab in last 5-7 years, and DST, which has given a couple of projects. ICR also, I started with an ICR project, and the team of students and my collaborators, which are all around India and abroad. So this is the last. Thank you.
Thank you, Bhim sir. It's a very nice presentation, and we were waiting for this particular talk because you have very good insight in this particular area, and you are doing very innovative work in this particular work, because you are not adding plant growth promotion, also you are adding very important information related with the biofortification and how can we add biofortification with the help of PGPR in particular plant. So I request Archie, do you have 1-2 questions, because
Yes,
Dr.
yes.
Bhim is having important meeting right now, so we will pick up only 1 or 2 questions for him. Yes.
Thank you so much, sir. So, Dr. Bhim Pratap Singh, sir, congratulations for this nice presentation. And we have questions from the participants. The first question we have, can fodder crops biofortified with micronutrients as in food crops?
Can
fodder crops biofortified with? Yes, sir.
Cotton crops can be fodder crops.
Yes, sir.
Yeah, fodder crops. It can be done. Fodder crops can be done, because at the moment among the biofortified projects, the first focus is on the cereals and grains. What the projects are ongoing. So till now, there is no project, I think, as per my knowledge, going on in the fodder crops, because they are targeting only the human consumption crops for the biofortification.
Thank you, sir. The next question we have, what are the major physiological limitations in cereals for micronutrients biofortification?
In cereals,
Yes, sir.
what are the
major physiological limitations in cereals for micronutrients biofortification?
cereals limitations? Actually, when we talk about biofortification, we are talking about micro nutrients like iron, zinc, provitamins. So limitations are not availability of these. Some of the not only cereals may have, but some of the crops, some of the foods which do not have these micronutrients, because cereals, see, they are it's not. I mean, what I can say that cereals may have some of the micronutrients, but availability of all the micronutrients which are needed for the growth and development can be checked, and if it is not there, we can develop biofortification for that particular cereal.
So thank you so much, sir. That's all the questions we have for you today. Over to you, Dr.
Yeah, so now I must thank you to Dr. Bhim Pratap sir for taking too much pain to be here because I know you are very busy right now. And
No, sir. Okay.
so thank you for giving a very important time from your busy schedule, and hopefully we will meet very soon in next program in the future.
Sure.
And
Sure, sir. Sure.
thanks a lot, sir. And we would like to collaborate with you for some work related with endophytes because some of my students are doing same work here.
Sure, sir. If we can do some biofortification, then it's a nice work.
Thank
we
you,
have
sir.
all facilities for doing the food processing, and this nano sciences also we are going to start a laboratory here, so we can do some nano biofertilizers or something.
Oh, very good, sir. So thank you very much, sir. Now it's time to leaving you. And
okay, thank you, everyone.
Actually, we have to start the next program. So thanks a lot, sir, for being with us.
Thank you, sir.
Thank you, sir. Thanks.